In these lectures, we will study different components of the cells in more details. Like in this tutor, are going to discuss about cell membrane in detail. Cell membranes. What is the cell membrane? Cell membrane may be outermost boundary of the cells. This is the outermost boundary of cells. We have already talked about it, and its functions as to boundary of the line of the cells. What about, let's talk about the structure of the cells. Structure of the cell membrane was given by two scientists like Jonathan Singers and Nicholson's Garth Nicholson's. They give the fluid mosaic model of the cell membranes. According to this model, it is consist of membrane bilayer and phospholipid bilayers, in which the proteins are embedded inside and both outsides, and phospholipid composed of hydrophobic tails and hydrophilic heads, like this ones. This is a phospholipid molecules. There are many phospholipid molecules, and they has polar head and non-polar tails, and their non-polar tails are insides, like this ones, and their polar heads are outside and inside and facing the fluids of extracellular matrix, and also facing the fluid and water of intracellular fluid, and their tails are against each other's, forming uh, layers which is hydrophobic. Like this is the structure or fluid mosaic model of the plasma membrane. You can see it. These are the phospholipids. They are heads. They are hydrophilic heads, and these are their tails. They are hydrophobic tails, and this is inside cells facing to the cytosol, and these are outside of the cells facing to the extracellular matrix, or facing or attaching to the another type. According to the fluid mosaic models, it is composed of two layers of the lipid bilayer, like phospholipid bilayers, into which proteins are ingraded or embedded like a mosaic manners, and it is a fluid structure, like not rigid structures. Plasma phospholipid molecule are moving like a water, like a fluid, and in its the proteins are also moving as icebergs are moving in oceans. So it is a fluid model, not a static models. And we will study about different structure, like phospholipids. Phospholipid has a polar head and non-polar tail. They are attracted toward each other, and the polar head attaches with the water of both side, intracellular and extracellular. There are also cholesterol in between the hydrophobic layer, like these are cholesterols. They provide stability to the proteins and also provide insolubility for water molecules, and they also provide help in stability. Like membrane proteins are also embedded in it. Some protein are outside, they are peripheral protein. Some are inside, they are internal proteins. Some are embedded in it, they are integral protein, and some proteins are embedded inside, such that it travels from outside to the insides, and some protein are just embedded in outside, or some proteins are just embedded inside. They help in signal transductions. Some molecules of the carbohydrate are attached to the proteins, and some act as a signaling pathways and a messenger system for cells. So it help in various functions, and they perform various functions. And when carbohydrate are attached to the proteins, like this ones, they are called glycolipids. And when they are attached, carbohydrate, when carbohydrate are attached to the lipids, they are called glycolipid. And when they are attached to the proteins, they are called glycoprotein. Like this one is attached to protein, it is glycoprotein, and both these structures are called glycocalyx, and that is outside the cell membrane, and help as a recognition of particular type of cell, and help in fingerprinting. Like this is the structure of the cell membrane of a particular type of lipid molecule. Like this is our hydro our phospholipid, hydrophilic polar head, hydrophobic non-polar tails, and these are the cholesterols attached to the phospholipid molecules, and it is more fluid regions, like tails is more fluid regions, and heads like where cholesterols are attached is a rigid region, मतलब like means that steroid or cholesterols make the cell rigid, and absence of cholesterols make the cell more fluid. If we talk about glycocalyx, these are the glycoprotein and lipid molecules. All these glycoprotein and glycolipid molecules are called glycocalyx. Helps in fingerprinting for particular type of cells, and marker for particular type of cells. They are also called cell codes. They are fingerprinting for particular type of cells, and they are composed of these glycolipids, like carbohydrate. These are the carbohydrate chains, glucose chains attached to the lipids called glycolipids, and these are the attached to the proteins called glycoproteins, and both structures are called glycolipids, help in particular type of recognition of cells. And inside of the proteins, like this is cytoplasmic site and this is outer side of the protein. Inner side there are the proteins attached, and outer side are protein attached. Let's talk about different function of the cells, like they regulate the movement of material inside and outside of the cells, separate the intrinsic of cells, also separate the organelles, like cell membranes are not only present in as a plasma membrane outside the cell membranes, but also separate the organelles within the cytoplasm from one another. So they separate various organelles within cells, and they provide large surface area and where large chemical reactions are present, like mitochondrial membranes are the membranes for large number of particular type of reactions we will study later on, like fatty acid fatty acid oxidation sometime take part in matrix, but started in membranes, and oxidation of NADH also take place in like oxidative phosphorylation take place in membrane surface, and many chemical reactions are taking place at the membrane of cells. They separate the cell from one another side. They are the site of receptors, and they have ability to pass certain substances through it, and have ability to hinder some substances from passing through them, and this property is called selective permeability, allowing some substances to pass through the membrane and allowing other substances to not pass. We will study about movement across the cells. There are various movement of the cross cells are, and all of these movement occurs through the cell membrane we will study about later. And